Okay, so exercise C, we're going to look at the presence of proteins and amino acids. This first exercise, C1, is specifically on amino acids, and it's a ninhydrin test. That's the name of the reagent that's used. It is toxic, so if you were in lab using this, you'd have to use gloves, so it's not to get it on your hands. And we're going to test distilled water, leucine, the amino acid leucine, proline, amino acid, and then a mixture of liquid amino acids, and then glucose. Now, this case, what we use is a filter paper, and we would take the filter paper and fold it into quadrants, and you can see it and you can mark the quadrants as it is here. You mark it with a pencil to show where you're going to put the drop, and you want to make sure that the drop is discrete, that it doesn't touch the other amino acids present, right? So here, for example, you have water, you have leucine, and you have proline. And in this other one, we have liquid amino acids and then glucose. Now, before the color develops, it will look clear. And you wait for it to go inside the filter paper and dry, right, by putting it on this warmer table. And over time, the color will appear. And if it's got an amino acid, it'll be purple. The only exception is proline. Proline is the only amino acid that'll give you a yellow color. So that is positive, even though it's yellow. All the others, right, will be purple. Okay. Now, if it dries to clear, that's a negative. So, for example, here, glucose, you can see, doesn't have any color, so it's a negative. This is a mix of amino acids. It's purple. It's positive. Obviously, we're testing for amino acids. And so it's a very easy, down and dirty test for amino acids. So if you move to C2, in C2, we're testing for, it's a qualitative test for the presence of protein. Okay, the reagent in this case is Biuret's reagent. It's blue, just like Benedict's was blue in the first exercise. This also has copper, so it makes it blue. And this is toxic as well, so you'd have to wear gloves when you were working with it. And so we're going to use one of the little plates. The little plates, we're going to test albumin, water, glucose, skim milk, leucine, and proline with the reagent Biuret's. Now, this is, once you incubate it, what you're looking for is the presence of purple or lavender. So you want it to go from blue to purple. If it remains blue, that's a negative test. So you can see here, for example, water remained blue. It's the same color as the reagent was to start with, so that's a negative test. But you see here, there's milk, and it's turned purple, so that's a positive result. Albumin, this first one, also is purple, positive. So some of these are going to give you positive, and some of them are going to give you negative. Now, interestingly, these two amino acids, even though proteins are made up of amino acids, Biuret's doesn't test for free amino acids. It only tests for amino acids bound in a peptide bond. So despite the fact that proteins are consisting of amino acids, they will not be picked up on this test because it doesn't test for that.